Okay, in the third and final portion of Chapter 3, we're going to talk about lipids. And this is the one group of macromolecules that is not technically considered a polymer. So, despite that fact, there are some common subunits that get used to build a lot of the lipids that we're going to encounter in this class, and we'll see a couple of those subunits here in a minute. But lipids are really defined as being nonpolar, mostly made out of hydrocarbons, just hydrogen and carbon. And when we say something's nonpolar, that means basically it does not have any either partial or full charges. So these are going to be molecules that do not like to interact with water. They're going to be very hydrophobic for the most part. We will see some exceptions. Um, and, we're kind of conditioned to think of fat as a bad thing, and certainly too much body fat can be a problem health-wise, but there are tons of biologically important roles that lipids have, and you can see a few listed here, warmth, thermal insulation, basically electrical insulation around your nerve cells, water repellency. But really, the main place that we're going to see lipids in this class is in biological membranes. So, just to give you a few examples of some of these common building blocks that are used to make lipids, we have glycerol and fatty acids. Now, glycerol is a 3-carbon alcohol. I'll show you the structure here in just a minute. And then fatty acids are basically just hydrocarbon chains, so kind of long, straight chains of hydrocarbon. And we're going to link these pieces together with covalent bonds that are called ester bonds or ester linkages. So just like we saw special names for the bonds holding proteins and carbohydrates together, same story here, ester bonds. So here we're going to build a triglyceride, and you can see we're going to make this out of glycerol, which is that 3-carbon alcohol up here in the purple box. And then we're going to bond 3 fatty acid molecules. And we'll have a dehydration reaction at each bond, so we'll lose 3 water molecules in this case, attaching those 3 fatty acid tails, and we would end up with our triglycerides held together by these 3 ester linkages. And you may have heard of the distinction between saturated or unsaturated fatty acids. And basically, this comes down to the nature of these hydrocarbon chains in the fatty acids themselves. If they're saturated, like you're seeing here, that means that the carbon backbone is basically saturated with hydrogen. So it has the maximum number of hydrogens attached to all these carbons in the chain. And that is going to give you these straight fatty acid chains that compact together pretty densely. Whereas unsaturated fatty acids, they're not saturated with the maximum number of hydrogens. So instead, we will have one or more double bonds between the carbons in the backbone of that chain. So you hear about monounsaturated would be one where there's one double bond. This example here is a polyunsaturated, just meaning there's multiple, 2 in this case. But these double bonds between the carbons will introduce a little bend into the hydrocarbon chain. So this is going to prevent them from being able to pack together as tightly as the saturated fatty acids would. And so what this means is those saturated fats are going to tend to be the animal fats, things like butter and lard, all the delicious stuff, right? That's solid at room temperature. Whereas the plant fats or plant oils, vegetable oil, olive oil, for example, because of those bends in the unsaturated fatty acid tails, they're going to be liquid at room temperature because they can't pack together quite as tightly. Um, and, there's health consequences to eating lots of these saturated fats because they will tend to pack together and solidify in your circulatory system, right? Clogging up your arteries and restricting blood flow and causing heart attacks and strokes and all kinds of serious health issues. All right, so I gave you a short laundry list of some of the important roles that lipids have, but as I mentioned, the main place we're going to see lipids in this class is in the form of phospholipids, which are a key part of cell membranes. And in fact, we have a whole chapter coming up just about cell membranes, so we'll dig in a little bit deeper there, but I will show you the structure of phospholipids today, at least kind of set that up in terms of how they form a membrane. And then just a couple of other types of biologically important lipids I wanted to mention as well, and I'll show you an example of each of these, are some of the pigment molecules that you find in plants, so chlorophylls, for example, or carotenoids, which are absorbing light energy and being used to generate sugar in the case of chlorophylls. Those are actually lipid molecules. And in animals, a lot of the steroid hormones and vitamins as well that you may be familiar with are fats. Typically, they're modified fatty acids. So let's take a quick look at these. So here's the phospholipid that I mentioned is a key part of biological membranes. And the bottom half of the phospholipid should look pretty familiar here. You've got the glycerol molecule that we just saw in our triglyceride. But now instead of that glycerol being bonded to 3 fatty acid chains, it's just bonded to 2. And the third carbon then is attached to this phosphate group, which is also then attached to this chemical group called choline. Okay. And this has a unique structure because the bottom half of this molecule here is very nonpolar. That's what we would expect in a lipid. As I mentioned, that's usually what defines a lipid. So no charges is going to be very hydrophobic down here. So we call this the hydrophobic tail of the phospholipid. But then the phosphate and the choline groups, you can see, do have charges. So phosphate group has a negative charge, the choline group has a positive charge. So this, what's sometimes called the head of the phospholipid, is polar and is very hydrophilic, likes to interact with water molecules because of those charges. And you may remember water has those partial positive and negative charges. Okay, so the consequence of this is now if we put a bunch of these phospholipids into an aqueous environment, which our bodies are, they will spontaneously form what's called this phospholipid bilayer. And this forms because those hydrophobic fatty acid tails are facing each other trying to get away from the water, and the hydrophilic head domains are very happy to sit there in contact with the water. And I forgot to mention, but one term you should know associated with this type of structure where you have both hydrophobic and hydrophilic regions is this term amphipathic. So we would consider this an amphipathic molecule. And there other molecules aside from phospholipids could also be considered amphipathic as long as they have both hydrophilic and hydrophobic portions. So because of that amphipathic nature, when we put the phospholipids in water, they will form this bilayer. And this basically is the backbone of a biological membrane, a cell membrane, for example. All right, just to show you an example from those other two groups that I just mentioned, we got the carotenoids, some of these light-absorbing pigments. You don't have to memorize these structures, but here's one called beta-carotene, vitamin A. These are both very nonpolar hydrophobic molecules. Some of the steroids I mentioned that you're familiar with, so cortisol, cholesterol, testosterone. You can see they all have these kind of distinctive ring structures. I'm not sure why they left estrogen off of here, but it's got a structure very similar to testosterone. But again, very nonpolar. You don't see any charges here, so that's why they're defined as lipids. All right, so that is it for Chapter 3. In Chapter 4, we will talk about structure and function of nucleic acids, DNA and RNA.